This is going to be a fairly long video over the organic chemistry portion of the course to get everything you need to know for the rest of the semester in terms of organic chemistry. So first of all, organic molecules. We all know the word organic. We hear about organic foods all the time. This is a different use of the word. Organic molecules are molecules that have a backbone composed of carbon linked together as well as hydrogen. Okay, so organic molecules have carbon linked with hydrogen. And one thing about carbon and hydrogen is they share electrons equally. They both have about the same electronegativity, which means it is a non-polar covalent bond. So these bonds between carbon is non-polar, of course, because they're the same atom, but between carbon and hydrogen is a non-polar covalent bond. That means water and oil don't mix because oil is nonpolar, water is polar, therefore they don't really mix together. So you are composed of all kinds of carbon, organic molecules composed of carbon and hydrogen. So, and you also have a lot of water in you. So how does that work? Well, organic molecules can be made polar, and to do that, we add functional groups. Functional groups are chemical components that retain their chemical properties no matter where they occur. So some of them are polar, some of them form hydrogen bonds, so on and so forth. So they have different properties, and they maintain their chemical properties when they're stuck onto a large organic molecule. There are 7 functional groups important to the chemistry of life. We'll focus on a couple of them here. Hydroxyl groups. Basically, it's an OH stuck to the rest of the molecule. That R is the rest of the molecule, okay? An OH. Carboxyl groups. Notice a double bond between the carbon and the oxygen, and then an OH as well. Amino groups. There's a nitrogen here and 2 hydrogens. This is a very important one for proteins. Phosphate groups. There's a big phosphorus there. You should be able to ID that. These are the ones I'm going to hold you to for now. Know these. Be able to recognize these and know where they're found. They'll help you to identify larger molecules. So these functional groups are very important. They change the properties of molecules. We've talked about emergent properties before. Properties that emerge as you move up the biological hierarchy. Well, you have 2 very similar molecules here, estradiol and testosterone. One of them has an OH there. One of them you see a double bond to an oxygen here. Very small changes, and here's an extra group on this testosterone here, the CH3. Those little tiny differences don't seem to be all that important, but that's the difference between estrogen and testosterone, the hormones that lead to sex determination in humans. So there are a number of ways that organic molecules can vary in terms of their backbone. The length can vary. Notice ethane and propane. The position of a double bond, so where the double bond is, can vary. If there are branches, and also whether or not there are rings present, like this benzene ring that we see on the right side of the screen. So these are different ways that carbon backbones, along with their associated hydrogens, can vary from one molecule to another. Important to know those. Okay. The different structures will obviously have different properties. Now, another thing to keep in mind is that there are structural isomers that are possible here. So basically what we have here are structural isomers, like we see pentane here and 2-methylbutane here. They have the same number of carbons and the same number of hydrogens, so the same chemical formula, but there is branching involved. There are different physical structures. Same chemical formula, different physical structures. We also can have cis-trans isomers. A cis isomer, so let's say whatever the X is, it could be functional groups, okay? or the rest of the molecule. They occur on the same side. That's a cis isomer. And in a trans isomer, once again, the same chemical formula as the cis isomer, but in this case, the X's, which represent some other part of the molecule, are on opposite sides. So you should be able to recognize those. And then enantiomers, this is basically like a mirror image type isomer. It's the same chemical structure, but it's like your left hand versus your right hand. They have the same structure, but they're mirror images of one another. So these may not seem like major differences, but these cause massive effects as you move up the biological hierarchy. For example, some drugs that are same formula, but one's a cis and one's a trans, one will affect your body, one will not have an effect on your body. So I thought I'd kind of relate this lecture to the digestive system, something that you're all familiar with, where we have these different organs that break down different things, and then we utilize those raw materials in our own cells. So digestion starts in the mouth with saliva, and we chew things up, we swallow it, it goes to the stomach, acid is added, then that food, that bolus of food that we now call chyme, mixed with acid, makes its way to the small intestine, then to the large intestine, and then out of the body. And at each step, different things are digested, different things are absorbed. If we look at a nutritional label, we'll see most of the chemicals that we're going to talk about in this lecture. First of all, we see calories, which is something you should already be familiar with. In fact, these are really kilocalories, but a dietary calorie is actually a kilocalorie, and there's 90 of them here. We see fats, we see cholesterol, which is also a type of lipid with fats, sodium, an inorganic molecule there, but carbohydrates, including dietary fiber, and sugar. So we'll talk about those. Protein, that's another very important one. Things you don't see on here are things like DNA. They don't list how much DNA is in your food, but of course, all of your food has DNA in it because it was once alive. So let's talk about lipids first. So we see those listed here: total fat, saturated fat, and cholesterol on a dietary label. These include fats, waxes, oils, and chemical signals called steroids are also often composed of lipids. And these are not soluble in water because they're kind of like the pure hydrocarbon, right? You've heard of hydrocarbons like oil. They're just carbon and hydrogen for the most part. Lipids are mostly just carbon and hydrogen, so they're not water soluble because of those bonds, those nonpolar covalent bonds. If we look at an example here, this is a glycerol molecule, and here's a long fatty acid chain. Notice lots of hydrogens and carbons. And we can link this fatty acid to this glycerol, forming an ester linkage through a dehydration reaction. Hydrogen and hydroxide is given off, leading to a water being removed. That allows a covalent bond between this glycerol and this fatty acid called an ester linkage. It's a type of covalent bond. Here we have a typical fat molecule with that glycerol and 3 fatty acid tails. So this molecule is essentially nonpolar because of all these carbon to hydrogen bonds, nonpolar bonds. Okay? So if we look, carbon has an electronegativity of 2.5, hydrogen of 2.1. That's almost the same, so these bonds are neutral. They share electrons fairly equally. There is no charge, so they do not mix well with water. Remember, water has bonds between oxygen and hydrogen, which is very far apart on this scale, 2.1 and 3.5. That means oxygen is better at holding on to electrons. Oxygen becomes more negative because of that, leading to polar bonds. So therefore, water has a charge, but fats do not. Therefore, water and fats do not mix well. So one of the jobs of fats, of course, which is a type of lipid, is energy storage. There are 2 types of fats that you'll hear about: saturated fats and non-saturated or unsaturated fats. Saturated fats are like we saw in the previous slide. There's lots and lots and lots of carbons and lots and lots and lots of hydrogens, and all the carbons are filled up, meaning I'm just going to draw 2 carbons in this chain, okay? This one's going to be hooked to another carbon over here. But hydrogen, 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 and hydrogen, and we'll assume there's more carbon off this way as well. These carbons are holding on. They have 4 different bonds. They are maxed out. They can't bond to anything else. That is a saturated fat. They are saturated with carbons. An unsaturated fat would have a double bond somewhere in it, meaning these carbons are only able to hold on to 1 other hydrogen instead of 2. So this is saturated, this is unsaturated. Now, saturated fats tend to be solid at room temperature. Why? Because their long fatty acid chains are very straight. When they get cool, they get closer together with other fat molecules and they push tightly together. They compact very tightly. Unsaturated fats, however, tend to have tails. Because of those double bonds, it changes their structure. Instead of straight tails like we see here, the tails are crooked and they don't pack tightly together. So at room temperature, you still have a liquid. Think of olive oil. So saturated fats pack tightly together. Unsaturated fats do not pack tightly together, therefore are usually liquid at room temperature. So their chemical structure then causes an emergent property when you move up to the actual entire stick of butter here, and that the butter is solid, whereas olive oil is liquid at that same temperature. Okay, so that was just one example. I use fats as the major example of a lipid, but there are other examples as well. But I also want to talk about carbohydrates. So carbohydrates are, once again, carbon backbone, lots of hydrogens attached, and we have carboxyl groups and things. And when you drop them in water, these often form a ring. So they can be linear or they can form a ring, like we see here. And we number the carbons in the ring. So here's carbon number 1, 2, 3, so on and so forth. Let me see if I can get the laser pointer to work here. I guess it's not going to cooperate with me right now, but that's okay. Okay, so here's carbon number 1, 2, 3, 4, and 5. So linear form and, okay, hold on. Okay, linear form and ring form. So you'll often see them drawn like this. These are carbohydrates. This is an example of a very simple carbohydrate, a monosaccharide. This is glucose that we're seeing here. Chemical formula is C6H12O6. C6H12O6. There are 6 carbons, 12 hydrogens, and 6 oxygens. Notice there's a ratio here of 1. If I can get this thing to work, it's not behaving right now. 1 to 2. Oh, there it goes. Oh, okay, 1 to 2 to 1 ratio, carbon to hydrogen to oxygen. Okay, so we talked about lipids. Lipids don't form big long chains really, but a lot of these other molecules form great big molecules. So we have these small molecules like glucose, which we can call a monomer. Monomer, you see that word here? They link together to form a larger molecule called a polymer. So think of a monomer like glucose as a brick and a polymer like a brick wall. Monomers lead to polymers through dehydration reactions, which are reactions that give off water. So reactions that give off water are dehydration reactions, kind of like we saw when we formed a fat, where water is given off. Whereas if you break down a polymer, you need water, and that's called a hydrolysis reaction. You hydrolyze a molecule, you break it down. So macromolecules are built of monomers. They're long chains usually of monomers, and an example of a monomer is glucose, a type of carbohydrate. So one example of a monomer, just so you remember, is glucose. Come on, there you go. All right, glucose. And then an example of a monomer then would be starch. So monomer glucose, polymer, starch. So if we take two glucose molecules, or in this case a glucose and a fructose, both are monosaccharides. If we stick them together, we make a disaccharide, like sucrose, which is table sugar. We have a dehydration reaction, and that forms a type of covalent bond known as a glycosidic linkage. You should jot down which type of linkage is found in which of these molecules. So we had an ester bond, now we have a glycosidic linkage in the case of a carbohydrate. So dehydration, glycosidic linkage. Plants often use disaccharides as a form of transport because it's harder for a cell to digest this and to break it down through cellular respiration, so it's more easy to transport without being accidentally used by the organism. Examples are sucrose, lactose, and maltose. That's a disaccharide. Now, when we have more than two monomers, monosaccharides, which are the single rings of sugars, have more than two of those glued together, through a glycosidic linkage. Then we have a polysaccharide, meaning many saccharides. Lots of dehydration reactions forming long chains. An example of this is starch, often composed of alpha glucose. So alpha referring to the type. If you remember a little bit ago, we looked at a slide that showed different types of structures of molecules. So we had different isomers. Well, we have an alpha glucose here, which is an isomer of glucose, forms starch, which is used as storage for future energy uses. There's also cellulose, which uses a different form of glucose, a different structure there, and that forms beta glucose. So a different isomer called beta glucose, and that is involved. You might have some alphas in there as well, but beta is present in cellulose, which is what wood is made of. That's used for structure. Some organisms can eat cellulose, but most cannot. So there is energy there, but it's really hard to break down. And so those are examples of plant uses of these molecules, starch and cellulose. Meanwhile, in animals, we store energy using glycogen, okay, which is kind of a branched, very similar to starch type molecule made of glucose. Animals also do not make cellulose. We don't use starch. Instead, we have glycogen. Some animals, like insects, make a hard outer shell, their exoskeleton, out of chitin, which is as close as animals get to cellulose. Chitin is composed also of carbohydrates. So here's an example of the two different ways that this glucose chain, could form a ring. Two different isomers. We have alpha glucose or beta glucose. Notice it's a very similar structure. However, when you make a chain of alpha glucose, it looks like this. When you make a chain of beta glucose molecules, you end up with something that looks more like this. Not very different, right? Except the OHs alternate. The hydroxyl groups alternate in this chain, whereas they don't in the starch chain. So we have 1 to 4 linkage of alpha glucose molecules, or a 1 to 4 linkage of beta glucose monomers here, meaning the first carbon is stuck to the fourth carbon of the next molecule. This type of molecule here, very easy to break apart using digestive enzymes. However, these molecules are not easily broken apart with digestive enzymes. So cellulose is what dietary fiber is made out of. We don't break it down, it just passes through us. It's like eating wood. Whereas starch, we can easily digest into individual glucose molecules and then process those further. So here's an example of all of these glucose molecules stuck together. So we have beta glucose forming cellulose, which is what cell walls are made out of in the plant kingdom. Very hard to digest. That's where dietary fiber comes from. That's where wood comes from. Meanwhile, starch is composed of alpha glucose, and it's composed of amylose and another form called amylopectin, which is branched, both of which are not too hard to digest. In animals, we have glycogen, which is how we store some of our glucose for quick use, like in our muscles. When you need a burst of energy, we can release glycogen, break it down, and utilize it through cellular respiration to make ATP. So I'd like you to be familiar with, I'm not going to make you memorize amylose and amylopectin, but starch is composed of alpha glucose and also glycogen, which is the animal storage version of this. So not all of your sugar is stored as glycogen, just some of it. Some of it ends up eventually going into fats and other things. But glycogen is how we have how we store a glucose reserve for fight or flight responses. So we know cellulose has a lot of glucose in it. Does it have energy in it? Well, of course it does, but we can't eat it because our enzymes cannot break those glycosidic bonds between beta glucose molecules. Some organisms, like these termites, can break them down, or fungi can, especially bacteria. Bacteria, there are types of bacteria that can break this down. In fact, most of your termites can't break down wood, but they have organisms living inside them, and those organisms have bacteria inside of them that can break down those bonds, those glycosidic bonds, and actually digest the cellulose into glucose. And we know there's a lot of energy in wood because if there wasn't, wood would not burn, but wood definitely does burn. Okay, proteins. Proteins are 50% of the mass of your cells if you eliminate all the moisture out of them. Okay, we're mostly water, but 50% of the dry material is protein. Proteins are super important for living things. We'll see a number of reasons why, but it's part of your daily diet, right? Proteins are important, and one of the things they do is that they are the enzymes that control the chemical reactions of your body. Without them, you do not exist. Very important molecules. So some of the functions of proteins in your body, or in any organism's body really, once again, they are enzymes. An example is pepsin, which helps to digest other proteins in your stomach. Defense, in terms of antibodies that help you defend against invading organisms. Communication, so they are receptors. They can act as receptors. They can do a number of things. Receptors, excuse me, transport, so hemoglobin is an example. Movement, your muscle fibers, actin and myosin, are composed of protein. I mean, just think about it. You eat meat, which is muscle, to get a lot of protein, right? Well, if you're an omnivore. If you're vegetarian, you might eat beans or something that has a lot of protein. Structure, keratin, like in your fingernails and hair, that's an example of structure and proteins. And storage, for example, egg whites are pure protein that are, essentially pure protein there for food storage for a chick, for example, or whatever the organism may be. So lots of functions. So I would advise that you write these down, especially the jobs of the proteins. You are not as responsible for examples, except for real obvious ones like muscle and hemoglobin. So proteins are composed of amino acids. So amino acids are the monomers composed of a central carbon, and we have our amino group and a carboxyl group, and then we have a side chain, which we label as the R group, which varies from amino acid to amino acid. There are 20 different amino acids. Think of them like letters in the alphabet. You can make all kinds of different proteins by combining these amino acids in different ways. So the unique character of each amino acid is determined solely by that R group, because the backbone is the same in every amino acid. The R group is different. Those side groups, those side chains, determine the shape of the overall protein depending on where they are on the chain of amino acids. So, different types of amino acids. Nonpolar amino acids have R groups with a lot of carbons and hydrogens in them. So if you're trying to figure out the properties of an amino acid, ignore the backbone. I don't care that there's an OH here, that there's an O here. That does not make the amino acid polar just because there's an OH. You have to look at the side chain only. All of this stuff down here will be linked to other molecules. This is the backbone. It's the side chain that determines whether or not an amino acid is polar or nonpolar or whatever. So nonpolar amino acids have R groups, usually with a lot of carbons and hydrogens in them. Another type that's nonpolar are aromatic amino acids, which have R groups with an organic ring in them. While polar amino acids have R groups with oxygen or a hydroxyl group. So remember, just like in water, they share electrons unequally, making a polar covalent bond. Or if you have R groups that have acids or bases. You'll know they're an acid or base because when they are in water, they ionize and are either positive or negatively charged. Therefore, they're also going to react well with water. So here is an example of some backbones with hydrophobic side chains. Notice the side chains in yellow, all carbon and hydrogen, or they have a ring. Here are some polar side chains. We have a side chain, there's a carbon and a hydrogen, but also there's an oxygen there attached to a hydrogen. This would react, this would bond with water just fine. It would dissolve in water just fine. It would be in the presence of water just fine. It would be hydrophilic. Okay, this molecule over here, this has a double bond with an oxygen. This is going to be also hydrophilic. There is some carbon and hydrogen, but there's also an oxygen present. Oxygen being very electronegative, it will react fine. I mean, when I say react, I mean it will get along fine with water. It's hydrophilic. It's polar. Here we have an example of acids and bases. So when they hit water, they ionize. This one probably was an OH, but it hit water and gave off its hydrogen. Now it's O minus, O minus. So negatively charged side chains are acidic. Positively charged side chains are basic. And when you see that plus or minus, you already know they have a charge, which means they are hydrophilic. Why is it important to understand if these are hydrophilic or phobic? Well, they're all part of a larger molecule, a big protein. That protein, that areas of the protein that have hydrophobic parts will turn away from water, while the hydrophilic parts will turn towards the water, affecting their shape. So when we put different amino acids together, we have a covalent bond form through a dehydration reaction, forming a peptide bond. Think of Pepto-Bismol to help you remember that. Peptide bond. So we have a peptide bond form here. Water is given off. We have these different side chains with different chemical properties. So this one, sulfur, things with sulfur in them tend to be hydrophilic, whereas, and this one probably also hydrophilic. At least some of the time here. Let's see. It might be hydrophobic on this example. This one may be hydrophobic. These two hydrophilic. Okay. Anyway, in this example, we have the beginnings. We have what we might call a polypeptide chain, a chain of amino acids held together with peptide bonds. You get a bunch of those held together, and you start forming a protein. So when it comes to knowing on an exam which of these are hydrophilic or phobic, I wouldn't give you this one because we didn't really go into sulfur and what it does and all these. I'm going to give you examples. I'm going to ask you about carbon and hydrogen, whether there's an oxygen present, whether it's plus or minus. Okay, I'm going to give you ones that are fairly easy to interpret, like the ones I provided in these examples. Are they hydrophilic, hydrophobic, and how do you know? It's very important that you be able to answer how do you know. Not just that you memorized it, but how can you tell if you're given a new example you've never seen before? Oh, it's all carbon and hydrogen, therefore they share bonds equally. They have nonpolar covalent bonds. They have equal electronegativity. Therefore, they would be hydrophobic. That's an example of what I mean by how do you know. Okay, so you have a bunch of these amino acids lined up. Their backbones are joined through peptide bonds, and then you get this big, long structure of amino acids. This is known as the primary structure of a protein. Okay, then you have hydrophobic R groups and hydrophilic R groups causing that structure to fold. Not only that, but you also have hydrogen bonds forming between various parts of the backbone, helping to hold this protein to a specific shape. So hydrophilic parts and phobic parts are turning away or towards the water, and hydrogen bonds are forming between parts of the backbone and adjacent parts of this chain of molecules. So we had primary structure like we saw here. Now parts of this are folding one way or another, and maybe like this molecule is attracted to this one because its backbone shares a hydrogen bond with that one. Okay, that's how you get secondary structure of proteins. Primary structure is the order of amino acids. Secondary structure is folding caused by hydrophilic and hydrophobic reactions and hydrogen bonds between parts of the backbone. And we get two basic shapes here normally, an alpha helix or a beta pleated sheet. An alpha helix is a spiral-like shape, and a beta pleated sheet is like pleats. You know, it's pleated like a piece of corrugated metal or something. Then we fold this again and get a more complex globular protein called the tertiary structure. Then you take a couple of tertiary structures and you can stick them together to make a quaternary structure. So this is fairly complicated, but not too hard, not too hard. So when we're looking at tertiary structure, these are interactions between the R groups, the side chains that are holding this structure together. So hydrogen bonds, ionic bonds, disulfide bridges, which is a type of covalent bond that we haven't really talked about, but also van der Waal interactions, which are just slight attractions between hot spots. You know, hot spots, I mean slightly polar regions. Even on nonpolar molecules, electrons move randomly, creating slight hot spots, slightly charged areas, allowing molecules to stick together. This is how a gecko, for example, climbs a glass window with these van der Waal interactions. You can read about them in your textbook. And we also have hydrophobic interactions causing these hydrophobic molecules to turn towards one another because they're turning away from water. So an example of how this applies to humans, this you know, tertiary structure of molecules, curly hair versus straight hair. So if you have hair that you're trying to curl, for example, your hair is composed of proteins, of course, and those proteins can respond in different ways to different chemical treatments. So if you add a reducing agent to your hair, it'll break the disulfide bridges between the proteins in your hair and allow them to separate. And then you can reshape your hair. You can put a permanent in it, like a permanent wave. So you break those disulfide bridges, you curl it up, and then you add an oxidizing agent to your hair, which reforms those bridges, and then it holds that shape. So a heating iron you can use to curl your hair. It only breaks those hydrogen bonds, and it allows curls that last until your hair gets wet again. It doesn't make permanent curls, at least, you know, long-term curls. So hydrogen bonds reshape like that secondary structure of your proteins, whereas if you break the disulfide bridges and reform them in forming curly hair, that's changing the tertiary structure, which is it's a hydrogen, you know, one's a hydrogen bond, which is a weak bond. A permanent breaks and reforms disulfide bridges, which is a longer lasting hair curl. Not something I have much experience with myself. But it's all done by breaking and reforming chemical bonds so your hair holds a different position. I don't even know if people do this anymore. So if you have naturally curly hair, then your hair is curled and held together with these disulfide bridges. And if you want to straighten your hair, you break those bridges, straighten the hair, and then reform them so the proteins you know, reform their bonds and hold a more straight position. So that's all involving the keratin, the protein that's in human hair, changing the shape of those secondary and tertiary structures. Now, if you take two tertiary structures or more and you put them together, so these are completely separate polypeptide chains, separate primary structures that have formed tertiary structures, and you stick them together, you get a quaternary structure, such as hemoglobin. So you have this hierarchy of protein parts. Here's an example of a quaternary structure. You can see the secondary structure here. You can't see the primary structure, the individual order of amino acids here, but you can see alpha helices and beta pleated sheets here or there. I don't see too many beta pleated sheets in this one. Then we have a few other molecules stuck in here, this hemi group and this iron forming hemoglobin. So here's a great example of this in a diagram, but also showing you emergent properties if you change the structure slightly. So here's an example of the primary structure of a molecule, the order of amino acids. And this person, this is the order, and this person, the sixth amino acid here is different in this person. That doesn't seem like a big deal, right? But you fold this around to make a secondary structure, and they have slightly different secondary structures. You fold that around, you know, so we can't see the secondary structure here. We can only see the tertiary structure pictured. So secondary and tertiary structures are shown here. The tertiary structure has a slightly different shape to it. You put that together with other tertiary structures to make a quaternary structure, and you have a somewhat different looking hemoglobin molecule. Then you look at the organism. The organism shown above has normal red blood cells, but the organism with this shape ends up with sickle cell shaped red blood cells, all because one tiny difference in the primary structure of the protein. Primary structure causes a change in the secondary structure, which causes a change in the tertiary structure, which causes a change in the quaternary structure, which then causes, you know, a disease all the way up at the organismal level. But it's also an evolutionary adaptation. People with sickle cell, or at least who are carriers of the sickle cell gene, are more resistant to malaria. So we'll talk about that more later. Here's something to think about. Sickle cell disease is caused by a mutation in the beta hemoglobin gene that causes a charged amino acid, glutamic acid, to valine, a hydrophobic amino acid. Where in the protein would you expect to find glutamic acid? So think about that. Does it like water? Does it not like water? Would it fold towards water in a tertiary structure, or would it fold away from water in a tertiary structure? And so I'll let you answer that on your own. The last type of molecule in this talk are the nucleic acids, which include DNA and RNA, but also include ATP and other molecules that are important for life. And these are polymers of structures of monomers called nucleotides. And the major role of at least DNA and RNA is information storage. In the case of ATP, it's energy transfer. And in biology, a very important thing to note is the central dogma of molecular biology. The central dogma of molecular biology is that DNA codes for RNA, which codes for protein. Okay, very important. This is what your DNA does. It stores information that eventually gets coded as protein, which then determines the chemical reactions in your body, the structure of your body, all of these things that was originally stored in your DNA. So if we look at an individual nucleotide, something that you should be able to draw, okay? A phosphate group and a nitrogenous base are these things that are present, but at the core of this is a sugar, pentose, a 5-carbon sugar, and we number the carbons. This is carbon number 1, 2, 3, 4, and 5 is up here. It's going to be important that you learn that, which one is which, because it helps you to determine which end of the DNA molecule you're looking at. We number them. So attached to the first carbon is a nitrogenous base, which can vary from molecule to molecule. Attached up here to the 5th carbon is the phosphate group. The phosphate group and the the phosphate group and the sugar make up the backbone of the DNA or the RNA, and the nitrogenous base sticks off to the side, as we see here. This is known as the 3' end of the molecule because the 3rd carbon, which has a hydroxide stuck to it, is sticking off and not attached to anything else. This is known as the 5' end because the 5th carbon attached to a phosphate group is not attached to any other sugars. Okay, it's sticking off out here by itself. So this is like the head and the tail end of the molecule. This is how we determine which way the molecule is running, by which one of these carbons is sticking here unattached to another molecule. In this case, it's the 3rd carbon. It has a hydroxide group, but it's not hooked to another sugar. Here, the 5' carbon is attached to a phosphate, but is not attached to another sugar. So that's the 5' end of the molecule, the 3' end of the molecule. Those nitrogenous bases can vary. The pyrimidines are a single ring. You should be able to recognize a pyrimidine from a purine. Cytosine or C, T, so C or T in DNA, cytosine and thymine. And then in RNA, we don't have T. Instead, we have U for uracil. We'll talk more about that later. But there's T in DNA, but U in T in DNA, U in RNA molecules. And then the purines, which consist of 2 different rings stuck together, including adenine and guanine. You don't need to be able to recognize cytosine from thymine based on a drawing, but I'd like you to know that cytosine and thymine are pyrimidines as well as uracil, and adenine and guanine are purines. The sugars are almost the same in DNA and RNA. In RNA, we have ribose, which, if you look at the second carbon, it's attached to a hydroxyl group right there, right? If you look at DNA, however, it's missing that oxygen. There's just a hydrogen there. So this is ribose, whereas DNA is composed of deoxyribose. So DNA stands for deoxyribose nucleic acid. RNA stands for ribonucleic nucleic acid. Sorry, excuse me. Ribonucleic acid, essentially. Deoxyribonucleic acid versus RNA. Okay? So if you look at a molecule, if there's an H here, you know it's DNA. If there's an OH here, you know it's part of RNA. Let me go back. In this example, we didn't draw it in, so you can't tell what it is. But you should know, oh, if this had an H on it, you would know this is a monomer of DNA. If this had an OH drawn on it, you would know it's a monomer then of RNA. So that is important to understand. So it will help you if in your notes you draw these things. So you just make an easy drawing. It doesn't have to be perfect. You draw a sugar as well as you can, right? And right here you can put OH, and then you can say this is for RNA, right? Or H for DNA. This is going to be an OH, and you can number these 1, 2, 3, 4, and then up here is going to be number 5. All right, and this is going to be attached to a phosphate group, and this is going to be attached to a base. So there's going to be a phosphate group attached to this 5th carbon. There's going to be a base, which are these things over here. Right? These are the potential bases, which will be attached up here to that 1st carbon, and that will vary from molecule from monomer to monomer, what you find there, whether it's cytosine, thymine, adenine, and guanine in the case of DNA, or cytosine or uracil or adenine or guanine in RNA. So the major roles of these molecules: polysaccharides, these big sugar molecules, are used for structure and energy. Be able to recognize them, to be able to identify them. Also know the difference between a polysaccharide and a monosaccharide. Lipids are used for energy storage, communication, and they're parts of membranes. I didn't go into the membrane aspect, but phospholipids are a major component of cell membranes. Proteins have numerous functions. We had a whole slide based on the functions of proteins. And nucleic acids are for information storage, but also energy transfer in the case of ATP, which we'll talk about in a couple chapters from now. So that was that lecture in a nutshell. Read your chapter, do your guided reading questions, and if you have any questions, you can get a hold of me.